Among macronutrients, one essential nutrient is carbohydrate. Carbohydrates are organic molecules made up of three basic elements: carbon, hydrogen, and oxygen. And they are very, very essential to us. Carbohydrates like glucose is the simplest carbohydrate and it is needed for respiration. 5 carbon sugar like ribose and deoxyribose are needed for making nucleic acids of DNA and RNA. Cellulose is also carbohydrate that is required for making cell wall in plant cells. And similarly, starch and glycogen are the large carbohydrates that are stored within the cells for as higher reserves for glucose. When we talk about carbohydrates, so carbohydrates are of three types. One is known as monosaccharide. Mono means one, saccharide means sugar. Carbohydrates that are consisting of single sugar unit, these are known as monosaccharides. And the monosaccharides include glucose, fructose, galactose, and all these three molecules have got the same chemical formula, that is C6, H12, and O6. And these three molecules are isomers to each other. Other monosaccharides include ribose, needed to make RNA, deoxyribose, needed to make DNA. The main source of energy to us is glucose that is respired, whether aerobically or anaerobically, and this is glucose. If we eat on plant products, we obtain starch. If we eat on animal products, we obtain glycogen. And when this starch or glycogen is hydrolyzed within our digestive system, so they release glucose that is transported to each and every body cell, where cells take them and respire on this glucose. If we want to confirm whether our food does contain glucose or not, because the food that is sweet in taste is likely to contain glucose. So if we want to confirm that our food does contain glucose and not, so we can conduct glucose tests, and this is also known as Benedict test, or reducing sugar test, or simplest sugar test. So what we do, we take a test tube. In that test tube, we add 5 cm cube of sample solution that we are expecting to contain glucose, and we add 5 cm cube of Benedict reagent. Benedict reagent has got blue color, so when it is added to the test tube, so the whole mixture turns blue. So we heat that mixture in the test tube in the water bath, approximately at 90 degrees Celsius temperature for approximately 180 seconds, and we observe the color change. If after heating in the water bath at 180 degrees Celsius, it does not change color and the color of the test tube contents remain blue, it means no glucose, no reducing sugar. But if the color changes to green, it means very minute concentration of glucose available. If concentration changes to yellow, so it means it has got small concentration of glucose. And if the test tube contents gain orange color, it means high concentration of glucose. Or if they gain brick red color, it means very high concentration of glucose. So this Benedict test is not only the qualitative test, it is quantitative test as well. It ensures us that sample contains glucose if the color appears to be green, yellow, orange, or orange red. And these different colors also give us an idea of the different concentrations of glucose in the given food sample we have tested with the Benedict reagent. Then comes disaccharides, mean carbohydrates that are consisting of 2 sugar units, means 2 monosaccharide units chemically bind together to make a disaccharide molecule. A disaccharide molecule is the larger molecule. Like 2 glucose units will chemically bind to make 1 maltose molecule. And similarly, glucose and galactose will bind to make lactose molecule. And glucose and fructose can bind to make sucrose molecule. So sucrose, lactose, and maltose. These are the three disaccharides, and these have got the same chemical formula, that is C12, H22 and O11. And these three can be taken as isomers to each other. And when these disaccharides are hydrolyzed, they will be yielding the same monomers that they are made up of. Same monosaccharides they are made up of. Like when lactose that is found in the milk, it is hydrolyzed, it is broken down, it releases glucose and galactose. When sucrose is hydrolyzed, it releases glucose and fructose. And when maltose is hydrolyzed, it releases two molecules of glucose. Remember one thing, like monosaccharides, as monosaccharides are sweet in taste, water soluble and diffusible, so these disaccharides are also sweet in taste and they are also water soluble and diffusible. Now, among the disaccharides, maltose and lactose are taken as reducing sugars because when they are heated with Benedict reagent, they directly give positive result. But sucrose is not taken as the reducing sugar, rather it is taken as non-reducing sugar because when it is directly heated with Benedict reagent, it does not give positive result. So in order to confirm the presence of sucrose, we need to carry on a test that is known as non-reducing sugar test or sucrose test. This sucrose test, what we do, we take a test tube, we add 5 cm cube of the given sample solution, that is the sucrose sample, and we add 5 cm cube of dilute hydrochloric acid solution. We heat the mixture in the water bath near to boiling. Then we take out the test tube, cool it down to the normal temperature around, and then we add alkali 5 cm cube in that mixture. After that, we add Benedict reagent and we heat it in the water bath at 90 degrees Celsius temperature constantly for around 300 seconds and then observe the color change. Now, how these different treatments will affect, like heating the sucrose solution with the dilute hydrochloric acid, it will break down sucrose into its monosaccharide units like glucose and fructose. Treating the mixture with alkali will neutralize the acid, so that acid must not react with the Benedict reagent. And now the Benedict reagent will give us the positive result. If we have yellow color of the test tube contents, that means small concentration of sucrose. And if the test tube contents turn orange red or brick red, it means high concentration of sucrose. The third type of carbohydrates is polysaccharides. Poly means many, saccharide means sugars. So carbohydrates that are made up of many sugar units, these are known as polysaccharides. And polysaccharides are mostly flat in taste, means tasteless. They are insoluble, they do not dissolve in water, and they are indiffusible, means they cannot diffuse as well. And we have got the three well-known polysaccharides, that is starch, glycogen and cellulose. Remember one thing, when these polysaccharides are hydrolyzed, they are digested or they are broken down, they release the monosaccharide units they are made up of, like starch, glycogen and cellulose. These three polysaccharides are made up of glucose. So when they are hydrolyzed, they are likely to yield glucose. Starch is the storage molecule, higher reserve for glucose stored in the plant cells. Glycogen is the higher reserve of glucose stored in the animal cells. And cellulose molecule makes cellulose fibers that make up cell wall of the plant cells. And all these three carbohydrates, polysaccharides, are made up of glucose. Our food contains starch, that is the major source to us, and that makes the maximum bulk of our food. So this starch can be found in potato, wheat grains, rice grains, etc. So if you want to confirm the presence of starch, so we conduct iodine solution test, or this is also known as starch test. So what we do, we take a sample solution in a test tube and we add 1 or 2 drops of iodine solution. Iodine solution has got yellow brown or golden brown color. So when this is mixed with the starch solution, so iodine reacts with starch and it turns the contents blue-black. So blue-black color of the test tube contents is the confirmation that the sample contains starch. So this was the discussion about carbohydrates. We discussed about monosaccharides, disaccharides, and polysaccharides, and we discussed about their lab test as well that will help us to identify their presence and their concentration in the given samples. Now we proceed to lipids. Lipids are the organic molecules and they are also made up of three elements: carbon, hydrogen, and oxygen. Lipids have got very important functions within the cells, like membranes, including cell membranes. They are actually made up of two layers of lipids. Similarly, lipids present as adipose tissue, so they insulate the body. Lipids are required for making certain hormones, like there are three important hormones in the human body, like in males testosterone, in females estrogen and progesterone. These are the hormones that are made up of made up of special lipid molecule known as cholesterol. For certain vitamins, lipids are required, like vitamin A, vitamin D, and vitamin E. They are functioning well when they are dissolved in fats, when they are dissolved in oils. These oils and fats are two different types of lipids. Lipids are chief source of energy. Oils and fats can be respired in the body, and they can provide us with more energy when we compare with the proteins or the carbohydrates. Cuticle that is found on the surface of epidermis in case of leaves in plants. So this is also made up of wax, and this wax is actually a lipid. A lipid molecule is made up of three components. One is glycerol, that is a special type of alcohol, and it binds with three fatty acids. And then we have a lipid molecule, and this lipid molecule is also known as a triglyceride. A lipid molecule represents all the properties of the lipid it is a part of. So different lipids, like different oils or different fats, they have got different molecular structure that is different from each other. So when a lipid molecule is hydrolyzed, it will be broken down and releases three fatty acids and one glycerol. If we want to confirm the presence of lipids in a diet, in a given sample, so we will have to conduct the iodine solution test. But before going to the iodine solution test, there is another segregation of lipid that I would like to discuss. That is, lipids are generally classified into two types. One is known as saturated lipids. These are generally known as fats, and at room temperature they are found in the solid or semi-solid state, and they do contain maximum energies. And the examples are margarine, animal fats found in the animal body, or cream found over the surface of the milk, butter, or even ghee. So these are the examples of saturated lipids. They are generally known as fats. The second type of lipid is oils that are liquid at room temperature. They comparatively contain less energies, and these oils are mainly from the vegetable sources, like vegetable oils like canola oil is there, sunflower oil is there, corn oil is there, or even olive oil is there. So these are all the examples of unsaturated lipids that are known as oils. Oils can be converted into fats by the process of hydrogenation on the industrial scale. In order to confirm the presence of lipids, that is either in the form of fats or either in the form of oil, so we can conduct a test known as ethanol emulsion test. So what we do in that test, if we have the liquid sample, we take 5 cm cube of it, we add 5 cm cube of pure ethanol into that sample. We shake it well, and then we add 5 cm cube of the distilled water and shake well. What we observe, we see the test tube contents have turned cloudy and a foamy emulsion may appear over the surface. So this is the confirmation of lipids in the given liquid sample. But if we have a solid sample like peanuts, like almonds, like cashew nuts, or any other source, so what we do crush it first, grind it to powder, add that powder in the test tube, add 5 cm cube of pure ethanol and shake it till the whole powder is dissolved in the ethanol. Then we add 5 cm cube of the distilled water in the same test tube, shake it well, and we find the same results like cloudy test tube contents and foamy emulsion on the surface that confirms the presence of lipids in the given sample. Proteins, another type of macromolecules, macronutrients found in our diet, and these proteins are very important to us. These proteins are made up of 4 essential elements like carbon, hydrogen, oxygen, and nitrogen. And apart from these 4 essential elements, some other elements can also be found, but depending on different types of proteins. Proteins are very important because most of the enzymes, means all the enzymes that catalyze chemical reactions inside our body, within the cells, within the organelles, so they are all made up of proteins. Most of the hormones like insulin, like ADH, they are made up of proteins. At the same time, pigments like hemoglobin found in the red blood cells, that is responsible for the transport of oxygen. And similarly, chlorophyll, that is found in the mesophyll cells, in the chloroplast of the mesophyll cells in the leaves, that is also a pigment. So they are protein pigments. Without chlorophyll, there is no photosynthesis. Without hemoglobin, there is no oxygen transport. Similarly, these proteins means muscle proteins like actin filaments and myosin filaments responsible for muscle contraction. These are proteins. And we have proteins as storage molecules as well, like casein that is found in milk is the protein store, and albumin found, that is also known as egg white in the eggs, that is also a protein store. So proteins have got vital importance, and if we do not have any carbohydrate or fat reserves in our body cells, in our body tissues, so we can still respire on proteins, respire on amino acids. Just like glucose is the simplest unit of all the carbohydrates, so similarly, amino acid is the simplest unit of all the proteins. There are 20 different types of amino acids, and these amino acids can bind together in different number, in different sequence, in different ratio to give rise to different types of proteins. In our body, we can have a protein made up of hardly 9 amino acids, and we can also find a protein that is made up of more than 3,000 amino acids. But the type of amino acid used to raise different types of proteins is the same, that is the 20 different amino acids are there. These amino acids chemically bind together to make polypeptide chains, and these polypeptide chains can raise the larger protein molecules that serve various functions in the body. If we want to confirm the presence of protein in the given food sample, so what we conduct a test that is known as biuret reagent test or protein test. In this biuret reagent test, what we do, we take a test tube, we add 5 cm cube of the sample solution in that test tube, and then we add 5 cm cube of the biuret reagent. It is also known as biuret solution. This biuret reagent has got blue color, and all the test tube contents will turn blue. We will shake the test tube contents and we will rest it for a minute. And after a minute, we see the color change in the test tube contents. And if the test tube contents turn purple, that could be taken as purple, mauve, violet, or even indigo. Light purple color will reflect low concentration of proteins, and dark purple will reflect high concentration of proteins. But if color does not change, it remains blue, it means no proteins at all. Balanced diet. A diet that contains all the nutrients, and these nutrients are present in the ratios, in the proportions required by a living organism, required by a human. A balanced diet contains all the nutrients, and the nutrients are present in a specific ratio. And this specific ratio is desired or needed by that human, by that person. And this ratio, or you can say the ratio of these nutrients in the balanced diet, can vary from person to person, and this variation depends on lifestyle, age, and gender. Like a 25 years old male laborer needs more energy, more carbohydrates, more fats, because it has to do a lot of hard work moving things from one area to another area, compared to the same age, same gender office worker. So you can even say a 25 years old male marathon runner would be needing more energy than 25 years old male teacher who has got less activity level. So lifestyle can vary the demand of energy. Accordingly, energy yielding components must be present in the balanced diet. Similarly, when we talk about females, a 25 years old female needs more iron compared to 60 years old female. Why? Because 25 years old female is experiencing menstrual cycle, and she is losing blood in every menstrual cycle. In order to compensate for the loss of that blood, so she needs more iron in her diet compared to the 60 years old lady who is experiencing menopause and is not experiencing menstruation at all. When it comes to age, again, it makes a difference. Okay, means just the example I told you right now, so that is also reflecting the age as well, because 25 years old female is younger, is more active, and needs more energy and more iron compared to 60 years female. And even you can see that 25 years old female could be office worker, could be an athlete, and the 60 years old female would be most of the time spending her time at home. And even if we go with the gender, like 25 years old female would be needing less energy compared to 25 years old male, because males are more mobile, more active, so they need more energy compared to females. You can yourself experience that the playgrounds like football ground, hockey ground, or even cricket ground that are set for female players, they are even smaller compared to the ones that are set for the male players. So that is actually on their energy level. So the balanced diet, one thing I repeat, balanced diet has got all the nutrients, and the nutrients are in the desired proportions, and the proportions desired is based on the lifestyle of the individual, on the age of the individual, and based on the gender of the individual. But not everyone is able to maintain the balanced diet in his daily dietary routine. If we do not take a diet with the balanced proportion, so this diet is taken as malnutrition. So malnutrition refers to intake of a diet that does not contain the nutrients in the balanced proportion. So this is known as malnutrition. And malnutrition can be categorized into like undernutrition and overnutrition. Undernutrition means one or more of the nutrients in the diet, they are present in less quantities than they are required. And the persons using such diets, they will be suffering from deficiency disorders. In case if a person is consuming a food that contains less iron, so that person is likely to suffer from anemia. If a person is using a food that contains less vitamin C, so that person is likely to experience scurvy. If a person is taking less protein, that person is likely to suffer from kwashiorkor or marasmus. And then it comes to overnutrition. If a person is consuming a food that contains one or more nutrients in larger quantities than what he actually requires, So that excess intake of certain nutrients can also create problem, can also cause abnormalities to occur. For example, excess intake of fats and sugars in the daily diet will result obesity, will result heart attack, chances of heart attack will increase, chances of stroke will increase, and blood vessels are likely to get blocked as well because of excess fats traveling through those blood vessels. And these people are likely to suffer with high blood pressure. So excess intake of any nutrient in the diet that is taken as overnutrition, that is also one of the category of malnutrition, and that is problematic to life as well. So we all need to have a balanced diet, and we all need to check what are our energy requirements of the day, and accordingly we must set our dietary chart. But in certain parts of the world, people do not find food, proper food. Forget about the balanced diet. They find it very hard to meet their daily energy requirements, their daily diet. This is what we call famine. Means people are suffering from famine in certain parts of the world, like in certain countries, in Africa, certain parts of the Asia, and the areas of the world they are constantly experiencing wars like Syria, like Palestine. So in these regions, the people are facing a lot of difficulty to meet their dietary requirements. So these people are suffering from famine. What are the causes of famine? Consistent wars in certain areas will not make these people to grow any crops, to carry any businesses, to earn any salaries or wages or any revenue from their businesses. So they could be suffering from famine. Countries constantly in wars, so they are unable to maintain their financial reserves to feed their people. Similarly, natural disasters like floods, hurricanes, earthquake, so they also destroy the vast areas. They also destroy the crops and the agriculture lands, and again there is little food. Similarly, locust attack on the fields can also deprive us with the food. So natural disasters, wars, poverty, poor economic state of the country will lead to famine. And the areas that are striking famine, that are experiencing famine, have to rely on the food aid provided to them from certain countries of the world, certain capitalists of the world. And they do not provide them with the balanced diet. They provide them with the diet that can meet their basic requirements. Like in the famine struck areas, people might be having food, but food is not balanced. And likewise, they are suffering from deficiency disorders, because if their diet is deficient in iron, they would be suffering anemia. Is deficient in protein, they would be suffering from kwashiorkor or marasmus. So we need to secure health in these regions by providing them with the balanced diet. That in this segment of lecture we discussed about nutrients, their types, their significance. Thank you very much, and stay tuned for the second video of the same unit, same topic.